supervising 3 students currently, and 7 candidates are currently working him for their PhDs. So total number of PhD students are 10. His area of research is to exploration of DFT calculations and MD simulations in different areas. He also works on exploration of sea-based molecules like GO, graphenes and water remediation and drug delivery. He has published more than 170 research articles, preprints, and chapters. Total citations to his work is more than 2,700. H index and I-10 index credited to his work are 29 and 81 respectively. He's listed in 2% of researchers in world created by Elsevier and Stanford University, USA. So we have two people, two scientists who are all almost in the same cadre. So, sir, with this, I would hand over the session to my to Professor Prashant Singh and Tokir, and we'll call the first.
A very good evening to one and all present here. Myself, Alka, and I'll be presenting my paper on the topic Template Assisted Mesoporous Tin Oxide Based Gas Sensor for NO2 Detection at Low Temperature. I'm from GGS IPU. The outline of my presentation is this: introduction, methodology, results discussion, conclusion, and future scope. Now we know nitrogen dioxide is a very harmful and hazardous gas, and it causes severe dangerous effects to human health as well as to environment. It is brown in color with a pungent smell, and if you long exposure to human health causes like diseases such as asthma, bronchitis. Nitrogen dioxide mixes with the moisture and causes acid rain. It is a major cause for photochemical smog and ozone layer deterioration. So, in order to detect the NO2 gas, we require gas sensors. So, gas sensors are basically devices that detects the presence of a particular type of gas in the atmosphere. There are different types of gas sensors, such as metal oxide based gas sensors, optical gas sensors, electrochemical gas sensors, capacitance based gas sensors, and many more. Our work is basically focused on metal oxide semiconductor based gas sensors. They functions on the principle of resistance variation upon interaction with the target gas. First of all, the resistance is measured in atmospheric air, followed by the resistance is then in the target gas, and finally the sensor response is calculated. So for a metal oxide semiconductor gas sensor to be highly effective, it must be high sensitive. It should have a very quick recovery time, a very fast response time. It should be repeatable. It should show a good reproducibility, and it should be highly selective. So we have chosen mesoporous metal oxides as gas sensors because they have high surface area and porous structure, which enhances the sensing sites and accelerates the adsorption desorption process of the gas, and hence overall improves the rate of change of resistance, thereby improving the sensitivity. So my work includes tin oxide as NO2 gas sensor. We know tin oxide is an N-type semiconductor with a bulk band gap of 3.6 electron volt. Literature has shown that NO2 tin oxide exhibits a great sensing behavior for NO2 gas, but at a very high operating temperature ranging between 100 to 300 degrees Celsius, which is a major research gap. So in order to minimize this research gap, mesoporosity can be used. It is a very impactful technique which has shown the NO2 detection at relatively lower temperature. We have introduced mesoporosity in tin oxide sample by using soft template. We have chosen CTAB as the soft template, and this is the tin precursor we have used. And the CTAB was finally removed by the solution by differently washing it with different solvents, mixture of solvents, ethanol and distilled water, and by providing the thermal treatment. And this is our final product, mesoporous tin oxide. XRD pattern of synthesized mesoporous tin oxide was in concordance with the JCPDS card number 770447, and no extra peak was obtained. This all the patterns exhibited the tetragonal reticle structure of SnO2. The average crystallite size was calculated using Debye-Scherrer formula, and it was found to be 4.8 nanometer. The porous morphology of the sample was confirmed, was predicted from FESEM. We can see the pores in the structure. The TEM images showed that the synthesized sample had spherical shape, and the HRTEM images showed the lattice fringes at 0.33 nanometer corresponding to 110 crystal plane, and the average particle size calculated from the TEM came to be 4.9 nanometer, which is in corroboration with the XRD results. The mesoporous structure was confirmed by the end-to adsorption desorption studies. The BT surface area calculated was 154.4, and the average pore diameter calculated was 3.2 nanometer from BGH method. This showed the type 4 isotherm with H2 hysteresis, confirming the pores to be bottlenecked in shape. This is the FTIR spectra, and the major peaks of vibrational peaks at 459 and 529 centimeter. They correspond to the OSNO stretching and SNO stretching. No major other impurity peaks were present. The chemical state of the sample was determined by using XPS. From XPS survey spectra, all the elements present were tin and oxygen. The difference between this high resolution XPS two peaks, it was 8.4 electron volt, confirming the tin present in tetravalent state. Now, the sensor was made by depositing this powder on the thin films containing interdistant electrodes of platinum using spin coating. This is our final sensor which we prepared and tested against NO2. We used 10 ppm concentration of NO2. This was the entire gas sensing measurement setup. It included the Keithley resistance variation setup, and this was connected with the desktop on which the resistance values were calculated or were interfaced. This is the sample holder. Here we have placed our sensor. These are some of the basic terms which we require to know if in gas sensing. So this is the a graph A. It represents the resistance dependence of the sensor in air. We see that with increasing temperature, resistance goes on decreasing, which is a typical behavior of a semiconducting material. This graph B represents the dependence of sensor on interaction with 10 ppm of NO2 gas. This is the sensor response curve C. From curve C, we can see that we have the maximum response at 60 Celsius temperature. But if we see the response and recovery time curve, the recovery at the 60 Celsius is at relatively very high time as compared to 80 So we have chosen 80 as our optimum temperature, keeping in mind quick response time and quick recovery time and the high response. These are the response recovery curve of mesoporous tin oxide sensor. This is the repeatability curve. We have taken 3 cycles. We tested our sensor for approximately like 300 days, and we get that our sensor was able to detect NO2 gas each time. However, some of the sensor response was deteriorated by the time because of the collapse of, I guess, mesoporous structure. Okay. This is the selectivity plot. We checked the interference capacity of the gas sensor with different gases, and we found that the effect of other gases was negligible as compared to the NO2 gas. So our sensor was highly selective for NO2 gas at 80 Celsius. So coming to the gas sensing mechanism, as SNO2 tin oxide is an N-type semiconductor. When it comes in the close 7. Okay. This was the research work, and this is our present work, literature survey. So, in conclusion, I would state that the highest recorded response was 8365 of our sensor at an operating temperature of 80 Celsius, and the response time was 1 second, and recovery time was 165 seconds. Our sensor showed a great repeatability, reproducibility, and was highly selective at 80 Celsius. And the attainment of such sensing behavior is ascribed to its high surface area owing to mesoporous structure and small crystallite size of tin oxide. A future scope would be a synthesis of doped mesoporous tin oxide and its gas sensing performance towards NO2 gas. So I would like to acknowledge Guru Gobind Singh Indraprasth University for providing me the platform to conduct my research, all the central research facilities for sample characterization, and Miranda House for gas sensing apparatus. And I'm very thankful to my professor Vishali Singh to always encourage me and provide me full support. And I'm thankful to Deshbandhu College for providing me platform to present my work. Thank you. Thank you.
Any questions?
Alka, in your introductory slide, you have shown that the optical band gap of this tin oxide now was 3.6, and it shows N type of semiconductivity. What is the role of these actually studies in the gas sensing properties?
So, as the band gap is very important factor for gas sensing. So, if the band gap is very high, then this this conduction mechanism would be like slow. So, we need to slow down, deep down the band gap. We need to narrow down the band gap so that the conduction mechanism goes faster and sensor is able to detect the gas.
The band gap was high actually. How
So
did you
no, sir,
actually?
this was for bulk. So our nanomaterial,
If you go for nano,
huh,
band gap will increase or decrease?
so it will increase
Then
because the size of particles
and
slow.
you need to
So,
decrease the band gap.
but sir, other factors are also into consideration, like high surface area and particle size. So our surface area was quite high. So it provided a large number of active sites for gas sensing.
Which is the dominating factor, surface area or the optical band gap?
So in our case, surface area is the major factor.
Any more question? If there is no question, let us thank Miss Alka Singh. Yeah, please.
Alka, it's a good presentation.
Thank you, sir.
I have a query.
Yes, sir.
What is the criteria of selecting CTAP?
So CTAB is a surfactant, and we are mainly focused on introducing mesoporosity to tin oxide. Like we can do sensing. It has showed in literature, many researchers have sensed NO2 gas with bulk tin oxide, or simply synthesizing tin oxide by using tin
No,
precursor tin
my question
chloride.
is what is the criteria? Is it cationic surfactant, anionic?
It is cationic surfactant.
So why? What is the criteria for taking cationic surfactant?
So, because it like if we missiles formation, we are adding ammonium hydroxide to it, and then we are adding tin chloride. So it is cationic. So like ammonium.
Okay, one more question.
Hmm.
You have recycled your materials. You have done the sensing of NO2.
Yes, sir.
Have you characterized your material after sensing?
No, sir. That we haven't done yet.
Okay.
Like our film has prepared after, like, we have characterized all the powders. So after preparing a film, we have done sensing on film. So
Yes.
after preparing the film, up till now we haven't.
After performing the gas sensing
Nahi,
experiment,
sir, we
you have
haven't.
mentioned that recyclability, you have seen the absorption and desorption are
Yes,
very
sir.
good. But what happened to morphology of your film?
So that we haven't seen. We haven't characterized FSM
Okay.
after, like,
Thank you, sir.
we do that, sir. We do that. Okay, thank you.
Thank you, Miss Alka, for the nice presentation.
Thank you, sir.
Now, Yogesh Kumar is absent today. Now, yes, come. Oh, that's great. So I invite Mr. Yogesh Kumar to present his work. Your time is 10 minutes. Yeah, total time is 10 minutes. 7 plus 3. 7 minutes is presentation and 3 minutes is question answer. Mr. Yogesh will speak over synthesis, characterization, physiochemical study, and antifungal activity of novel choline based ionic liquids and their conjugates with fluconazole drug a vitro and vivo approach. Yogesh.
Good evening, all. Good evening, respected teacher. Hello. Good evening, respected teacher. I will present on the uh, synthesis, characterization, and antifungal activity of choline-based ionic liquid and with and it conjugate with fluconazole drug. And this is introduction. What is microbial infection? Microbial infection is that infection that is due to microbial, uh, due to virus, bacteria, and fungal and plasmatic infection. And type of fungal चल नहीं रहा ये मैम ऐसे बोल दो ऐसे बोल दो सर
उस पे खड़े होके बोलना है आपको.
type of fungal infection, uh, fungal infection, fungal infection, uh, joke itching fungal infection, and yeast fungal infection.
Hello.
Next is antimicrobial agent, and it is type. Antibacterial agent and antiplasmatic agent, antifungal agent, and antiviral agent. Type of antifungal drug. There are four type of antifungal drug, and azole, choline, and alinamine, echinocandid. Ionic liquid and their properties. What is ionic liquid? Ionic liquid is salts consisting in organic cation and inorganic anion, having temperature below than 100 degrees C. These have several properties like high non-volatile nature, green solvent, high solubility nature, low melting point, high thermodynamic stable, good ionic conductivity, good surface activity. Also have the application in pharmaceutical field, solubilization of drug, biosensing, drug delivery, protein stabilization. Now I coming on the my work, synthesis of choline based ionic liquid. This is the reaction involved in the synthesis of choline based ionic liquid. Firstly, we prepare the choline bromide using the ethanolamine. Secondly, you use the uh, synthesis of choline prolate using the choline bromide ionic liquid. Uh, this is the synthesis of ionic liquid drug conjugate. This is the reaction involved in the synthesis of the ionic liquid drug conjugate. Uh, Next part is the characterization. We characterize with the this is FTI NMR physiochemical. We do the physiochemical characterization using the surface tension measurement and compute the different surface parameter like CMC, pi CMC, tau max, A minimum, PC70. They are the equation used in the for computing the parameter. And surface parameter is support with the different technique, UV visible fluorescence and conductivity, and uh, we obtained a very sportive result with the surface tension measurement result. Biological activity, mechanism action of the antifungal drug against the fungal strain. Disc diffusion assay. This is the MIC study we obtained uh, in the drug, ionic liquid drug formulation give the lower MIC value as compared to pure ionic liquid. It suggests that uh, after the formulation of ionic liquid drug formulation give the best result against the fungal strain. This is the gross curve assay, MTC assay. Here we obtain the ionic liquid and drug ionic liquid drug conjugate. show the low toxicity against the HEK-293 cell line up to desired concentration. This is the conclusion.
Yogesh, why you have taken only choline chloride based ionic liquid?
Because choline based ionic liquid is less toxic as compared to imidazolium pyridine based ionic liquid.
Okay. So how did you find the presence of presence of moisture? You have synthesized the choline based ionic
Yes.
liquid.
Yes, sir.
My question is there any possibility for intercalation of water or moisture from the environment in the ionic liquids?
Y- Yes, ionic liquid gain the moisture from the environment, but for every experiment we do the uh, we use the hot oven to remove the water moisture.
No, no, this is not my question.
जी जी sir.
If I wish to know how much moisture is present, I'm talking about intercalation of water in the ionic liquid.
I have no idea.
Okay. So please work on it.
क्या सर?
एक बार इसको पढ़ने की
जी
कोशिश
जी सर
करना.
जी सर.
Any more question? Thank you, Yogesh. Yeah. So I think the next presentation is by Subham Tiwari. Subham, yeah, are you there? Yeah. Good. Subham will speak over nucleophilic sulfur-driven kethoamide synthesis by making nucleophilic amine.
Good evening to all. Today I'll be presenting my current research work that is entitled as Nucleophilic sulfur-driven ketothiamide synthesis by masking nucleophilic amine. In this work, we'll be discussing on the synthesis of alpha ketothiamides. So alpha ketothiamides are a very useful class of organosulfur compounds used in various fields of studies like organic synthesis, pharmaceutical, as well as natural products as well. So they found themselves useful in building blocks because various heterocyclic compounds can be derived from the ketothiamides, as well as various organosulfur compounds find themselves useful in the fields. Besides organic synthesis, they also found themselves as a promising candidate in the pharmacological industry as well, as some of the ketothiamides, quinolone based ketothiamides, are known to show antibiotic properties, while some others are known to show anti-cancerous activity by regulating the activity of phosphoglycerate dehydrogenase enzyme, which is responsible for the tumor cell formation inside the human body. So when we go through literature survey, there are various methods to synthesize ketothiamides. Majorly, these are divided into three major categories. First, which make use of aryl methyl ketone or alpha functionalized ketone with elemental sulfur and amine to get the alpha ketothiamide as a product. Next methodology uses alpha beta enaminones or alpha beta unsaturated carbonyl compounds with elemental sulfur and amine to get the ketothiamide as a major product, while some other uses oxidation of the aryl thiamides to get ketothiamides. So why there is a need to develop a new methodology is because when we go through literature search, we found that all these methods are having certain limitations, like some of them are using dry reaction conditions. And some of them are using expensive chemicals, while some of them are using long reaction time. Harsh condition is there in some cases, while the most common thing which is there in all the existing methodology is there is a requirement of pre-existing carbonyl center within the starting material. So we thought to devise a method which eliminates these limitations, and we get ketothiamide as a major product in a greener approach. So we started our work and we took tribromomethyl carbinol as a model substrate and treated it with elemental sulfur and amine, and we got ketothiamide as a major product. In this, what are the advantages of our method is that no pre-existing carbonyl center is required in the starting material. Reaction goes under aqueous condition. No transition metals are used in our study. reaction does not take very long time. Condition is free from harsh reaction conditions as well. And also cheap and stable starting materials are used for the synthesis of final product. So when we go through our work, we also found some challenges, like besides getting ketothiomide exclusively, we also got thiomide as a side product during our study. So we have done extensive optimization of the reaction conditions to get ketothiomide as a sole product. First of all, we did reaction in presence and absence of water. In both the cases, reaction was going. However, reaction profile was more cleaner when we used aqueous conditions. Later, different equivalents of sulfur and different sulfur sources like sodium sulfide and sodium thiosulfates also we screened, and elemental sulfur was found to be most promising candidate. Afterward, we screened different solvents like acetonitrile, DMF, methanol, ethanol, and DMSO, and THF was the most best solvent for this transformation. Afterwards, we screened this reaction at a different temperature, and a temperature of 65 degrees Celsius was found to be optimum for this transformation. Also, different equivalents of amines also we have screened, as the amine equivalent could be the major culprit for thiamide side product formation. Also, we have screened different amount of additive so that ketothiamide can be formed in a better yield, but no further improvement in the yield was observed. And we took our standard condition as a to screen substrate scope. So a broad variety of substrate scope we have screened, which include electron withdrawing groups, electron neutral groups, as well as electron donating groups as well. It is important to mention that two compounds which are synthesized by our protocols are reported to show anti-cancer properties as well. Besides getting this substrate scope, we also did this reaction successfully with bifunctional groups and also polycyclic, heteroaromatic, as well as aliphatic substrates as well, and corresponding products were formed in good to excellent yields. Afterward, we have screened this reaction and we have showed the substrate scope with different divergent amine, and a wide variety of divergent amine gave corresponding products successfully, like secondary amines, primary amines, and all gave the corresponding product in good to excellent yield. Also, the ester derivative of amino acids was also compatible and the corresponding product was formed in excellent yield. Further, ketothiamide having dimethyl substituents were also compatible and products were obtained in good yields. However, with some bulkier amine, the reaction was not compatible and no product was obtained as complex mixture we achieved during the reaction mixture. So besides getting the ketothiamide as a sole product, we also got thiamide as a limiting amount in some cases, which indicate that the optimized reaction conditions are sufficient enough to limit the formation of thiamide side product. When we screened this reaction with 2-fluorotribromomethyl carbinol as a model substrate, surprisingly, we got the fluorine was substituted by the amine counterpart and the corresponding product was formed in a good yield as a major product. Afterward, to prove the reaction mechanism, we had carried out different control experiments. Like first of all, we have treated the tribromomethyl carbinol with elemental sulfur and no reaction was observed. Second, we carried out the reaction of tribromomethyl carbinol with the amine and a new spot in the TLC was observed. We isolated that and characterized that, and the intermediate was found to be an aldehyde, which was isolated and characterized by NMR spectroscopy. Later, to prove the intermediate is aldehyde only, we have carried a next experiment in which we treated tribromomethyl carbinol with morpholine, and in the reaction mixture itself, we added elemental sulfur, which gave exclusively thiamide as a major product, which indicate that aldehyde was the major intermediate during the reaction. So after literature search and control experiment we carried out, we proposed a tentative mechanism for this transformation in which elemental sulfur and amine first reacted together to form the polysulfide ion 1, which reacts with the tribromomethyl carbinol to generate intermediate 2, which after removal of HBr leads to intermediate 3, which after ketoenol tautomerism and further, sorry sir, yes sir, got the product in a major yield, while the nucleophilic attack of amine to the starting material gave us aldehyde, which is a major product which we characterized in our control experiment, which on reaction with the polysulfide delivered the thiamide as the side product. Also, we have showed the synthetic application, scale up reaction and synthetic applicability was shown. So, in summary, we have developed a new protocol to synthesize alpha ketothiamide, which utilizes readily available starting material. Moderate reaction temperature was required. Reaction goes under aqueous conditions. Also, synthetic application was demonstrated by scale-up method and further synthetic transformation as well. So finally, I would like to acknowledge my research supervisor, Dr. Guru Raja GN, for providing me research facility, and Central Instrumentation Facility, CUG Gandhinagar, for providing me analytical support, all teaching and non-teaching staff, all my lab members. And also, I would like to thank ICHE for providing me the podium for presenting my research work in front of you. Thank you very much.
Any query, guys? You might have interest in organic chemistry. You may ask. Okay. Shubham, your presentation is interesting, but I have some queries. In organic reaction, you have used amine, an aqueous solution of amine, and you have used THF as a solvent. So both of them have different polarity. Why you have done so?
No, sir, we have carried out this reaction in presence and absence of water because as an organic chemist, we do not use water as a solvent in our study. So for greener approach, we have used water as a solvent besides the co-solvent THF.
So that I want to know.
Yes,
What
sir.
is the role of using two solvents?
Means
They are not miscible in each other.
no, sir, THF and water are almost miscible to
Miscible.
each other. Yes, sir.
So you should run the reaction in any of them.
But sir, reaction yield was more better when we used both of them together.
Okay.
Also, by using water, we can reduce the amount of THF also, so that reaction can go by greener way. Means solvent use can be limited.
Okay. One more thing which I like to ask. In one of the product you have shown
Yes, sir.
stereochemistry. So how did you find the stereochemistry? Can you go back to that slide?
Sir.
One of the product had stereochemically active. Go back. Yeah, this one. How did you find this stereochemistry?
So actually, it was L-leucine methyl ester hydrochloride as a amine part. We have treated this means L-leucine methyl ester. It was itself R configuration.
But how did you find? Did you?
No, sir. It means configuration. We have not changed the configuration of the means chiral center. It is not affected. Means that is only amine reacting. Means if the amine is reacting, the configuration won't be altered. Otherwise, if we will synthesize a new compound to check the configuration, we have to do XRD.
What you have to do?
By XRD, we will know whether it is R or S.
Okay. One more thing which I like to ask. How did you find the yield of each product?
Sir, when we done the reaction, we have passed the reaction mixture through column chromatography, and isolated products we weighed and got the yield.
Can we determine the yield by some other technique?
Sir, by
The more
HPLC,
reliable and applicable technique by which you can say the major product is of 70% and the minor is of 30%.
sir, by means in some control experiment, I have seen that they are reporting percentage yield by using NMR only.
So did you perform NMR for this?
No, sir. NMR we can get yield. The means most of the papers are using column chromatography to get the
After then you weighed the compound.
Yes,
and
sir.
you
Yes,
said
sir.
there is possibility these are amines, and if you have used silica,
Hmm.
there is a possibility that amines will bind on silica.
Yes, sir. That is why
So
we have.
how did you find it's not possible that all the compound is eluted out?
No, that is why, sir, we used to write isolated yield in that case as a major compound. And besides your question, that amine may stuck to the acidic column, we have passed the silica through 2% of ethyl acetate solution to neutralize the silica so that our compound won't
Neutralize
get.
the silica.
Yes, sir. Neutral silica we have used to do column chromatography.
Uh, okay, thank you, Subham. The title slide actually should carry the name of the supervisor, right? You only just thank that the facility is only provided by the supervisor. Actually, the idea is also from the supervisor. Admission, your admission is from the supervisor. Everything is from the supervisor. So your title slide along with your name, है ना?
Yes,
The
sir.
supervisor's name should carry it.
Okay, sir. From
है ना?
next time I will.
Okay.
Thank you, sir.
So thank you, Subham. So the next presentation is again by another Subham. Subham Kumar. Yeah, Shubham Kumar. So Subham Kumar will speak over the systematic molecular modeling study to explore the nature's arsenals against Alzheimer's disease.
Hello. So, good evening, everyone present here. My name is Shubham Kumar. I'm currently pursuing I'm, I'm currently pursuing BTech from BIT Meshra और I will be presenting topic on the system. अच्छा मेरी आवाज आ रही है पीछे तक आवाज आ रही है okay so today I'll be presenting my topic on systematic molecular modeling study to explore nature's arsenal against Alzheimer's disease. Okay, so first of all, what is Alzheimer's disease? सुना तो होगा सबने कि भाई एल्जाइमर्स हो गया, सिम्टम्स सुना होगा, मेमोरी लॉस वगैरह, लेकिन एग्जैक्टली होता क्या है? बेसिकली एल्जाइमर डिजीज dedicates that it leads to it's a neurodegenerative disease where the neuron actually degrades over the period of time. It is a very quasi static process. So जब भी हमें पता चलता है हम ऐसा assume करते हैं कि हमेशा ओल्ड लोग में ही होता होगा, लेकिन it is not compulsory. So in case of Alzheimer's, uh, it is characterized by progressive loss of function and structure of neurons in the brain and spinal cord. तो Alzheimer's actually होता कैसे है? What is the pathway? When you look at the right side of the this frame, the amyloid beta. This is amyloid precursor protein. It was supposed to be cleaved by alpha secretaries and then follow its normal enzymatic property. Whatever the proteins are supposed to do. But due to some unknown reasons, unknown factors, when it's cleaved by beta secretases and gamma secretases, this leads to an improper cleaving. This causes formation of amyloid beta peptide, this one right here. These amyloid beta peptides oligomerizes, aggregates, and then forms amyloid beta plaque. Now this plaque gets jammed between neurons, and due to this, the Alzheimer's occurs. Am I clear? Any doubts? Sir? Is it clear? Okay. So, now what is the effect of Alzheimer's on the structure of the brain? You can see the right side of the brain, the cortex shrunk and the ventricles grow to fill up the spaces that the cortex has created. The spaces are filled by ventricles. The ventricles grow larger and are filled with cerebrospinal fluid. The Alzheimer's usually take place in 3 stages: the early, middle, and later. Now, our research basically involves the inhibition of the amyloid beta peptide aggregation in the early stage itself. So, now what about the drugs that are actually available? No drug yet gives a pure, perfect cure of Alzheimer's disease. Mostly the drugs that are available and approved by FDA give symptomatic relief. Like one of the drugs, aducanumab. You can see here how it's its effect over the period of 1 year, but it is highly criticized by scientists regarding its effect and amyloid beta aggregation. They say that it's harmful. So we can say that there is no perfect cure regarding the amyloid beta, regarding the Alzheimer's disease, though no perfect drug is available yet. So in my rationale says that we have already seen through the years of publication, through research, that uh, products derived from natural sources, products derived from natural sources such as curcumin and resveratrol, these has shown to prevent the formation of amyloid beta plaques, hence to protect brain from Alzheimer's disease. Not exactly Alzheimer's disease, other dementia and neurodegenerative disease like Parkinson and I think it's called Huntington's. Yes. Okay. So we in our research will employ two computational techniques. First, molecular docking, and then molecular dynamic simulation. Now, what is molecular docking? Molecular docking states that, okay, the image is not very clear, unfortunately. This is one peptide that I showed, that pink color one, that orange color one, and suppose, and no, not suppose, actually this is another peptide. Now, if in case of Alzheimer's patient brain, the oligomerization of these peptides leads to a diseased neuron. Now, through molecular docking, we can find one ligand which comes in between. That is the receptor pocket, which was supposed to join these two receptors together. It's now blocked by ligand. So due to this, there is no aggregation and hence a healthy neuron. Okay. Now regarding the, now I have been continuously saying peptide, peptide, peptide. Now what about the structure of peptide? First, this peptide, this here. This is the classical, like traditional fibril peptide of amyloid beta, which I have taken from protein data bank. It's used for traditional studies, and uh, like the conventional, when we say that we have to study computationally Alzheimer's disease, this structure is taken. But the issue with this structure is that the amyloid beta peptide, it is actually an intracellularly disordered protein. That is, in case of tumor cancer and other proteins, the structure of the protein structure is definite. Whereas in case of amyloid beta, there is no like rigid or definite structure. It changes over the period of time. So we took fibril structure as shown in the last slide, fibril structure from protein data bank. This structure we have created using molecular dynamic simulation of simple peptide and through clustering process. And we have one more structure of PTM. PTM have shown, especially phosphorylation serenade has shown to accelerate the amyloid beta aggregation. Now these are the rankings showing the docking process of our ligands. We used a library of 307 ligands and screened them, uh, then perform molecular docking for each cases. Now you can see why three structures we have to take. Niastatin, in this case it was top. For S2 cases it's at bottom. So if we perform docking of only one case, it may lead to that we might miss another important compounds in this case, in this case. So that's why we need to perform molecular docking for all three cases. Now after that, we found 7 common compounds out of those 307. In top 20, we found these 7 compounds. Now we further perform their ADMA studies and consider factor like Lipinski and good bioavailability and whether they crosses BBB, blood-brain barrier or not. After that, these are some 2D interactions and their 3D structures. You can see how beautifully the rascogenin is binding with each protein structure. Now, why molecular dynamics? Sorry, sir. Yes, sir. So you can see the initial structure of 6 peptide. Now, these are the motions that peptides take place. These simulations are conducted over a period of 100 nanoseconds. You can see how the peptides are aggregating over the period of time. That's yeah, that's the final structure of amyloid beta peptides. These gets jammed between neuron and then causes Alzheimer's disease. Now, since we are on time crunch, I have to explain the MD simulation results quickly. So these are 3 systems which took. One we took control, six peptide, 3 are rascogenin. Okay, sir. So yeah, these are the graphs that I obtained, and it shows positive results that you can see our ligand uh, get gets between these two peptides and basically jams their aggregation. So we can thus conclude that yeah, our ligand is a good drug candidate for potent drug development, and it can be concluded through in vitro and in vivo experiments. Uh, this is acknowledgement for our research group, Advanced Computing Lab. He's my PI, Dr. Alugyan sir, and Ms. Asta Tiwari, who helped me to perform molecular docking studies. So yeah, thank you, sir.
Any queries, students, participants?
Hello, hello. Yes, sir.
What is the resolution of PDV taken?
Resolution, I mean, it's like as I said earlier, the peptide amyloid beta peptide form. Okay.
I'm asking, did you download the PDB from RCSB?
Yes, sir. Obviously.
So what is the dimension? What is the resolution of this PDB?
What is the resolution of the PDB?
Yes.
Sir, sorry, sir, didn't get your question.
Okay. So further, please tell me, how did you prepare your protein for for performing molecular docking and MD simulations?
Yes, sir. So for molecular docking, we first took the structure, we deleted water molecules from it. We added polar hydrogens and gassteric charges.
Through which software?
Autodock Vina. Basically, we used the protein preparation to Autodock tools, and we performed docking through Autodock Vina.
Okay. So in Autodock, how many poses you obtained?
9. There's a cutoff actually. You can take how many poses you want, but I took 9.
Okay. So the one problem is observed by most of the researchers.
Yes, sir.
That the results of molecular docking are not reproducible.
Yes, sir.
May I know the reason?
Sir, because as I stated, because of time limitation, I was not
No, you please respond to this query.
Yes, sir. Because the consideration in molecular docking is that protein is static. Okay, protein is protein is static. It assumes that protein is static, ligand static, and it's binding statically. Whereas when we consider MD simulation, we are also considering conformations of the period of time, like how the protein is moving, how the ligand is moving. So you don't seem convinced, sir.
Okay. So did you perform PBSA calculation here?
Sorry, sir.
PBSA calculations,
No,
because
sir.
you have used
No, sir. No, sir. Not yet.
Gromax
Not yet.
for AMD
Yes, sir.
simulations.
We use Gromax.
So please calculate the thermodynamic parameters.
Yes, sir.
for
Yes,
it.
sir. Yes, sir. That's the future aspect. We have to calculate thermodynamic parameters, and much more analysis needs to be done.
Okay.
Yes,
So
sir.
do you have any idea about PCA analysis?
Principal component analysis, no?
Yes.
No, sir. No, not yet.
Okay. Thank you.
Okay. Thank you, sir.
Next is Priya Yadav. Priya Yadav.
My name is Priya Yadav. I am from Dr. Bhimrao Ambedkar University, Agra. I am going to represent my topic on evaluation of toxic trace element in fine particulate matter and their influence on human health, environment. These are my objective contents. These are my objective introduction. In this, we all are aware about particulate matter. Particulate matter are suspended particles in air, which are very in shape and size. Basically, I am going to talk about trace elements in particulate matters. These are many toxic trace elements which can cause many harmful diseases to human beings and also causes monumental effect and environmental effect, crop disease. This is my work plan. This is my sampling site, Nunhai. Basically, it is a industrial area and near historical monument Atmad Uddaula. This is my work plan. By this method, standard operating method है. By this I collect my sample of PM 2.5. This is the sampler by using I collect my samples, and this is the filter paper I use. By gravimetric analysis, I calculate the concentration of PM 2.5 particles. By chemical analysis, I calculate the elements concentration in PM 2.5 sample. First, I digest the sample filter paper by the method of aquaresia or open vessel method. By ICP and by ICP-MS, I characterize the metals. This is my observation table. Uh, this is my study on 3 months in Agra. On my side, this is the concentration and also meteorological parameters. Mass concentration at my site, it is average concentration is 2.3 times and 3.5 times higher than the daily average and annual average of NACS. This is element. These are characterized in my study, in which iron, magnesium, aluminum, lead. These concentration of these elements are higher than other elements which are harmful for human health. This is my conclusion. In this, the concentration of PM 2.5 particles are higher than the annual average concentration and daily average concentration, which is prescribed by NACS and WHO also. Detected element by ICP-MS are which are very harmful for human health, and these these also affect the monument of at that site. It was observed that high traffic density, industrial emission, and biomass burning have greater influence on PM 2.5 concentration and on its elemental concentration, because at the site there is many ferrous and non-ferrous industries, and their localities is slum and densely populated. They are using traditional fuels for local activities. An unappreciated level of PM 2.5 at site in Agra. Mitigation should be implemented to minimize emission rate, and environmental impact assessment study is recommended to investigate the long-term impact of PM 2.5 particles. Thank you. These are my references.
Yes, Priya. First of all, I congratulate you because you have finished your PPT presentation within 5 minutes.
Thank you, sir.
So I have not reminded you that 2 minutes are left or your time is over. So I have only one query in your presentation. You have done ICP-MS.
Ji, sir.
So what is the percentage of the elements? Because you have shown an order,
Ji, sir,
that
ji, sir.
this is the order, and the percentage. What is the percentage of those elements?
Uh,
Can you go back to that slide? Conclusion. Left. Please help her. Conclusion. Yes, this one. Second point. So you have given an order. My query is, what are the percentage of these elements?
Iron, mostly iron percentage, which is 40%. Then magnesium, then aluminum, and iron. Exactly percentage I'm not remember the actual percentage.
But you should provide the percentage or amount or
Concentration
whatever.
amount
Concentration
provided
should be
as well.
given.
I'll
Go back.
say back.
So from where you get this ICP-MS analysis?
IIT Delhi.
I did Delhi. Okay. Oh, wonderful.
Consumption amount I provided, but percentage actually data बहुत dense हो रहा था. That's why I not use show the percentage.
No worries. Okay. Thank you. Next, Ravina.
Good evening, all of you. I'm Ravina, presenting my research work, which is based on the nanocellulose gallic acid metal organic framework. This compound is used for the food packaging material. First of all, we have used plastic in the many forms for carry bags and food packing materials, etc. These plastic bags are non-biodegradable, and it cause to the health and as well as environment polluted. First of all, when we dump the plastic waste on the water bodies, it pollutate the waters, and when we drink the water, that it goes in our body in the form of microplastic, as well as it polluted from the soil for the soil, and when from the soil it goes to the plants, and from the plants it goes in the um, food materials, and when we eat the food materials, the plastic goes in our body, and it cause many health it affect the health, such as the liver toxicity, digestive toxicity, reproductive immune problem, and cancer risks also. For this problem, many scientists has worked on the biopolymer based food packaging material, because this is easily biodegradable and low cost and abundance in natures, and this easily sustainable process to prepare these compounds. So, for example of biopolymers, the chitosan, which is used to the food packaging material because of the it has a high film forming properties. That's why the chitosan have used to the forming of the film, and it is easily biodegradation in the soil, and it doesn't affect the environments also. So many scientists has worked on the this chitosan based film was used to the food packaging materials, and for the materials activity will enhanced by the incorporation of the nanocellulose and bioactive compounds, because the nanocellulose have high mechanical chemical properties and high functional properties. That's why it easily interact to the chitosan, as well as bioactive compounds has antimicrobial antioxidant properties, which that's why the active film activity will enhanced by the synergistic effect, and it is easily biodegradable, and not cause any harmful effect of the environment, and also antioxidant and antimicrobial property as so. But it has some disadvantage, such as it not stable at high temperature, as well as low barrier and mechanical properties, and careful use to prevent degradation. To remove all these disadvantages, we have used the metal organic framework by using the bioactive ligands such as gallic acid, because the metal organic framework has high adsorption property, high surface area. That's why the metal organic framework, I have synthesized the metal organic framework by the simple hydrothermal method, and it was characterized by the IR spectra, and OH on the COH functional group was used to um, used coordination bond with metal ion, and it was characterized by XRD technique, and this XRD pattern is similar to the MIL53 rhombic structures, and the morphology of the material was characterized by the TAM images, and the TAM images spherical shape structure was observed. And after that I have synthesized the nanocellulose by the wheat straw, and it was oxidized by the tamper, and there's a CH2OH convert into CO and negative CO minus Na positive, and it was confirmed by the conductometer titration, and also it was characterized by the IR and the FESM images. In the SAM images it was looking like the nanofiber structures, and then I have synthesized the chitosan based film having nanocellulose and copper gallic mauve have different concentration. It was characterized by the FESM IR spectrum. In the IR spectrum, the OH peak and other peaks was decreasing in concentration. That's why we can say the uh, OH was interacting OH functional group through the hydrogen bonding of nanocellulose and MOF, and the it was stability was enhanced after the incorporation of the MOF in the chitosan film, as well as the UV your percent transmittance is also decreased after the incorporation of the nanocellulose and metal organic framework, and moisture content, water solubility, swelling degree also decreased by after the incorporation of the nanocellulose and metal organic framework in the
Last minute.
chitosan based film.
Last
And
minute.
though the it has so a high oxygen barrier property because the iodine value is uh, similar to the marketed oil. That's why we can say that the film is has high oxygen barrier properties. So that's why after incorporation of the nanocellulose and metal organic framework, all property will enhance, and that's why we can say the it will help in the food packaging material. And acknowledge. Thank you, sir.
You must read the acknowledgement, na? You must read the acknowledgement. What was your acknowledgement?
My supervisor, Dr. Pratibha Kumari, and also CSIR for giving me this opportunity, and
You
ICH.
should actually properly acknowledge the presentation,
Okay, okay.
right?
Sorry, sir.
Just say, okay, this is my acknowledgement, thank you. Huh? Okay, that is the
Time
important
is
side.
that's
No,
why.
no, no. We will give time from our side. Na?
Okay, sir.
Okay.
Sorry.
So it was a good presentation, Ravina.
Okay, thank you, sir.
Come back to the HRTM slide. Can you go back? Go back. This one, 7 number. 7 number. Next. Yeah.
Yes, sir.
So, which one is the HRTM image?
Yes, sir.
Which one?
Both are TAM images, sir.
Both are TEM. Which one is HRTM?
HRTM is 2
Hmm?
2 nanometer. Okay.
2 नैनोमीटर जिस पे लिखा हुआ है 0.2 नैनोमीटर लिखा हुआ है
हाँ,
वो।
नीचे वो 2 nanometer है.
अच्छा, okay. So how did you index this HRTM image? Question is this. How did you index this HRTM image? Did you get my question? You have written, you have draw two parallel lines, right? Right. You have written D equals to 0.2 nanometer,
हाँ.
right?
Yes, sir.
How did you analyze this portion?
By images software, the
No,
interplanar
this is not from
distance.
image software. This is done by you only.
हाँ, this जो distance is measured by the images software, and this is the interplanar distance, sir.
Then this 0.2 nanometer, did you compare with anything or any interplanar distance or any plane?
No, sir.
What D equals to 0.2 nanometer signifies?
Yes, sir.
What it signifies? क्या बता रही है ये चीज? क्या बताती है? D आपका 0.2 नैनोमीटर आ गया, ठीक
Yes,
है ना?
sir.
आपने उसकी थिकनेस निकाल ली है,
हाँ
आप
जी,
बोल रहे हो इंटरप्लेनेट डिस्टेंस की वो है,
हाँ जी,
डी वैल्यू है
हाँ,
नैनोमीटर में।
हाँ।
अब इससे क्या इन्फॉर्मेशन मिली आपको, बस ये बता दो।
अच्छा,
ठीक है, तो आपने
इन्फॉर्मेशन।
दिखा दिया, इन्फॉर्मेशन क्या मिले? पीछे जाओ जरा, एक
सॉरी।
स्लाइड पीछे जाओ। ज्यादा पीछे चले गए क्या? 6 पे जाना था, पीछे जाना। 6 स्लाइड, स्लाइड नंबर 6।
Yes,
See the
sir.
X-ray diffraction,
Yes, sir.
right?
Yes, sir.
Your D value is 2 angstrom,
Yes.
0.2 nanometer, right?
हाँ जी, हाँ जी.
Then compare with the D value with the X-ray diffraction, okay?
Okay.
And the particular D value having a corresponding plane means HKL. So
Mm-hmm.
on the basis of the lattice fringes, right,
Mm-hmm.
you predict the corresponding lattice
Okay.
plane of that particular material.
Okay, sir.
Okay.
Okay, sir.
Now come to TGA plot.
TG plot.
Next. 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 I think 13 or 14.
Next. हाँ. इससे भी
Next.
next.
नहीं आगे चलते हैं.
Hmm.
Yeah, this one.
Yes,
This
sir.
one.
Yes, sir.
Your x-axis is percent weight, right?
Yes, sir.
And sorry, y-axis, and x-axis is temperature.
Hmm.
In all the plot, your weight loss coming to 0.
Yes, sir.
Is it possible?
Percent weight loss.
You started your measurement from 100 weight percent.
Yes, sir.
After certain temperature,
Hmm.
it cuts the x-axis, whatever the weight, no? But coming actually touching to the
5.
x-axis.
Starting point
There
from
must be
5,
some
sir.
stability, ना कुछ तो बचेगा. हाँ. What is your end product actually in these reactions?
End product of the
हाँ.
reaction. I don't know, sir. End
अच्छा,
product.
ये टीजीए करते क्यों हैं? क्या इन्फॉर्मेशन लेते हो आप?
बेसिकली टीजीए के कितने टाइम तक, मतलब कितने टेम्परेचर तक कंपाउंड स्टेबल है, उसके बाद वो मेल्ट हो जाता है. कम हो
नहीं, अगर मान लो चलो वो मेल्ट भी कर रहा है
हम्म.
तो 0 वेट पे आ जाएगा. वेपराइज होगा तो 0 पे आना चाहिए. वेपराइज हो जाता है आपका पूरा, ऐश भी नहीं बचती है.
सर बेसिकली
चलो कोई बात नहीं, आपने शायद खुद नहीं किया ना, कहीं पे कराया है.
हाँ कराया है सर.
ठीक
हाँ
बात.
जी.
ओके, एनी मोर क्वेश्चन?
One query.
यस सर.
What structure you expect for this move?
स्ट्रक्चर्स. हाँ, हाइड्रोजन बॉन्डिंग इंटरेक्शन है सर.
Here you have taken some cellulose
हाँ जी.
based molecule. One metal is also there, copper.
हाँ, मेटल ऑर्गेनिक
So what
फ्रेमवर्क.
structure you expect? Because nowadays people are working on DFT calculations for
ओके.
MOF
ओके.
for a particular application as also.
ओके.
So we should have a structure
of
for any
metal
MOF.
organic framework.
Yes.
Yeah. This is reported metal organic framework, and it has a rhombic structure.
No, I'm asking about structure, how it look like, how it appears.
Look like.
Like benzoic acid, we know COH over phenyl,
Huh.
molecular structure.
Molecular structure of metal organic framework.
Yes.
Gallic acid use the ligand, and the um, copper metal ion coordinate with the bond of the COH.
But you should show the structure because you
जी.
have also prepared it.
हाँ जी, हाँ जी.
Then structure must be shown in your presentation.
Okay, sir.
Thank
Sorry,
you.
sir. Thank you, sir.
So thank you, Ravina. Do we have any more presenter? Any more? Yeah, Jyoti Yadav, Ankit Sharma, Sagrika Sharma. Sagrika is here? Okay, yeah. So we have the last presenter, Miss Sagrika Sharma. Yeah. Sagrika, you have 10 minutes, 7 plus 3.
Okay. Good evening to one and all. My name is Dr. Sagrika Sharma, and I'm here to present before you my work on strategic portfolio management in drug development. So I got the opportunity to work with a professor in Spain when I was studying in Yada Business School, and I have been able to gather some points that I would like to share here. So this is the table of contents. We'll be talking about the overview of portfolio management in the pharmaceutical industry, the multidimensional approach to portfolio management, what can be the implementation strategies and the approaches that we can follow ahead. So to start off with, we need to understand what is the current scenario in the pharma industry. As we know that the pharma industry has been growing immensely and it is quite dynamic in nature. But we have been able to understand that the patent has a shelf life. The patent only exists for a period of 20 years, after which the expiration, the companies find out different ways to enhance their patent life. This can include reformulations, this can include getting into orphan drugs or rare diseases. Apart from that, they've also been dealing with loss of exclusivity of their blockbuster drugs. One of the example of an MNC is Pfizer, and Pfizer has dealt with this with the cholesterol-lowering medication, which is Lipitor, and we will be discussing about it briefly in the upcoming slides. So the dynamic decision making depends on how do we deal with this situation. As you can see in the graph here, a lot of companies from 2019 to 2025, it is predicted that have experienced loss of exclusivity. And they have been spending this in brand spend, in having a new emerging brand. So the loss of exclusivity of these drugs, along with the finite patent life, also poses challenges when it comes to long developmental times of these drugs. As we know that these drugs take about 9 years to 12 years for its development from the initiation until the market. We need to also understand that it comes in various phases. We have the R&D phase and then the clinical trials phase until getting into the market. When we talk about the R&D, as we try to identify the compound, we have hit to lead optimization and beyond. Then we come into the animal trials, the animal clinical studies, preclinical, and the clinical trials 1, 2, and 3 phase, and then we are able to register ourselves for an approval and get into the market. But the story doesn't end here because all these are associated with very high costs. Also, there is a possibility that there is across the stages of 1, 2, and 3, there is a possibility of success and a probability of success that is calculated, which is based on 70%, 33, 25 to 30%, and 90%. Apart from that, all the drugs that are started from initiation, out of 21, only one is able to enter the market, which is alarming in nature. So not only it is expensive, but a lot of fund allocation is required at this end. And it is important to have a very strategic approach. So what can be our strategic approach for a corporate growth? To be able to have an R&D project strategy, a portfolio strategy, a technology strategy, and be aligned with the values and mission and vision of the company. We are trying to identify how we can be beneficial by understanding the NPV, the ROI, and all the technical feasibility and commercial success. We are able to understand the internal dimension, the strengths and weaknesses of our own products, and how we can manage them effectively. So this is just a brief of the project portfolio matrix that talks about the technical feasibility and the commercial feasibility. And just to have a very simplified version, we have put it in 4 forms, which is the bread and butter, pearl, oyster, white elephant, which just talks about the net present given success and how it can maintain its values in terms of competition and uniqueness when it comes to technical feasibility. Apart from that, there's a very famous quote which says, fail early, fail cheap. That talks about if a drug has been failing at the earlier stages, it is very important to identify it and manage the other drugs effectively. So if we are trying to create more options, it is important to understand and develop the drugs that will enter into the market depending on the current scenario. Moreover, to balance the resources that we have and to allocate the funds effectively. So for this, we need to have a very multidimensional approach. We talks about strategic alignment. That is, do the strategic goals align with the drugs that are getting into the market? Are we able to manage the risk, that is the clinical trials failure, all the regulatory changes that comes into the picture? Are we able to allocate our resources? Do we have the enough infrastructure to handle it? Are we able to diversify, or are we diversifying it way too much, depending on if it's a small or a big size pharma company? What is the performance measurement? And if we are able to monitor it for a long term, are we able to prioritize and terminate and select the drugs that are into getting into the market or may or may not succeed? What will be our optimization and how we'll be able to adjust it? And how do we tackle the stakeholders? Because stakeholders are the ones that are most important. These can be our regulatory changes, that could be our patients, and all the dignitaries that are involved. So this is just a small brief analysis which talks about what would be the link to strategy, so that we're able to establish a clear guidance and optimize the existing portfolio to be able to balance our portfolio and have newer projects into the market if needed. But also look if our strategy is concise and if high level review is needed. A robust portfolio would also include identifying the sweet spot and have a strategic value, customer value, and investment intensity. To be able to establish this, we need to understand that apart from all these points that we have talked about, there is also called scoring models that we assign values to different categories as per risk, cost, or the feasibility as per our own categories to be able to establish the next process. Financial matrix, this is just an example of how the projects say this is the portfolio of all the drugs that exist. What would be the probability of success and how we can forecast it to be able to achieve a better reality. To be able to implement, we also need to look at the value story, which talks about the unmet need, what is the efficacy, the safety, what would be the economic burden, and the benefits if the drug gets into the market. Moreover, as I talked about it in the previous slides, so Lipitor had been able to establish its presence by an acquisition and merger, by alliances and partnerships with a company called Vaith in 2009. And that's how it diversified not only its portfolio, got into biologics and also into vaccines, but it was able to emerge as a winner because it entered into emerging markets, so it could offset any distances that it had in its revenue at that moment of time. And this is one of the examples of how expiration of blockbuster drugs can lead to newer innovation and newer partnerships ahead. This is the summary of how we can establish clear goals and lead into success of our drugs. Thank you so much. I would like to thank the podium and the ICHE conference for giving me this opportunity to present my work ahead. Thank you.
Any question from the audience? So if there is no question, let us thank Sagrika
Yeah.
for the nice presentation. Good luck.
Thank you so much, sir.
So madam, now over to you.
Thank you, everyone. Thank you, the audience, and thank you, Professor Taqir Ahmed and Professor Prashant Singh, sir, for giving your invaluable insight and inputs to the students. Hope that they will incorporate in their work. I would like to now, this is the end, so I would like to invite now Dr. Umesh to kindly felicitate Professor Taqir Ahmed with a memento as a token of our appreciation. Thank you, sir. Now I would like to invite Dr. Archana to kindly felicitate Dr. Prashant, Professor Prashant Singh, with a memento as a token of appreciation.
and I must say that all the participants have presented very nicely.
So this session ends over here. And after this, we'll be having a cultural program very soon in the auditorium.